Hello everyone, welcome to the fourth day of the web series on health and diseases contemporary concept. Today we have with us Theodore Price from Center of Advanced Studies, University of Texas at Dallas, USA, in conversation with Dr. Murmun Chattopadhyay. This presentation was organized in collaboration with National Academy of Sciences, India, Indian Indian Association of Neurosciences, and Institute of Life Sciences, Bhubaneswar. This is a part of our Ravenshaw 150 Distinguished Lecture Series, where we invite people from around the world to share their experiences and enlighten us with their life's journey. With these words, I welcome Dr. Rakesh to introduce Professor Price to you. Dr. Rakesh.
Thank you, Professor Luna. It's a pleasure to introduce Professor Price. I'll start with the lines of Greek physician Hippocrates. He said that from the brain alone arises our pleasure, jest, sorrow, grief, as well as pain. However, in ancient culture, religion influenced belief about pain. Having a pain symptom was regarded as God's punishment. But with the evolving human thinking, medical technology advancement, pharma, industry expansion, and use of animals as experimental pain models suggested that actually pain is an unpleasant feeling associated with tissue damage. Sense of pain is important because it warns an individual of existing medical problem that has to be addressed well. Thus, pain protects us from a severe injury and a serious medical problem. During ongoing COVID-19 pandemic crisis worldwide, Pain Alliance Europe, it's a group of 19 countries in Europe, conducted a survey on a chronic pain patients from UK, France, Denmark, Sweden, Finland, and Netherlands, and they found that SARS-CoV-2 infection aggravates pain condition in these patients. So, therefore, besides taking COVID-19 precautions, it is very much important to avoid any injury or damage. Soon we are going to be enlightened by an interesting talk on pain by Professor Theodore John Price, where he will be sharing with us his journey on signs of chronic pain. About Price, he has a lot of achievement. I'll be highlighting some of them. He is a professor at the Center for Advanced Pain Studies at University of Texas, Dallas. He is a Bachelor in Neuroscience from University of Texas, Dallas, and in 2003, he completed his doctoral study in pharmacology from University of Texas Health Science Center, San Antonio. In 2004, he joined Professor Fernando Rivero lab at McGill University for postdoctoral research. In 2007, he joined as assistant professor at the Department of Pharmacology, University of Arizona, Tucson. In 2014, he returned to University of Texas at Dallas as associate professor, and currently he is a professor at the School of Behavior and Brain Sciences. So he is recipient of several research grants, awards, and honors, and I'll just mention some of them. He has been honored with our Science Program Award for Excellence in Science, IASP Patrick DeWall Young Investigator Award, Eugenie McDermott Endowed Professorship in 2018. He is a member of ISP Review Committee. ISP is an international organization which organizes by every second year International Congress on Pain, where neuroscientists from entire world meet together to discuss the progress and their promise for the pain research. And he is also a committee member in peer review committee, and he is a member of American Pain Society Board of Directors. Professor Price is actively working in area of pain for 20 years, and he has more than 100 published research articles. He is using behavioral pharmacology, molecular genetics, electrophysiology, and single-cell transcriptomics to explain how activity-dependent translation in nociceptors regulates plasticity in spinal dorsal horn neurons. His investigation has now grown into a field of study that is yielding important new mechanisms and targets for other pain research laboratories involved. His work highlights the role of protease-activated receptor 2 and AMP-activated protein kinase as a novel target for the treatment of neuropathic and post-surgical pain. Despite improvement in health infrastructure as of today, millions live with pain and dies in pain. So we need to take pain seriously, and we need to address these two, because the first symptom a patient reports in OPD is pain. So we need to address this symptom well. So now let us hear from Professor Price his point of view. And now I would like to invite Munmun Chattopadhyay, who is my senior and assistant professor at Texas University Health Sciences Center, to have conversation with Professor Price. Ma'am, Munmun ma'am, please.
Hi Ted, good to see you.
Good to see you too.
So I know you for a long time, but I would like to introduce to my side of the world. When did you study? Think of what motivated you to study neuroscience during your undergrad. I believe that's the time.
Yeah, that's true.
Neuroscience. So what motivated you?
I actually always wanted to be a physicist, and
Wow.
yeah, so I was. I went to University of Texas at Dallas. I'm from Dallas. I was actually born in Kansas City, which is right exactly in the middle of the United States. But my parents moved to Dallas when I was a little kid, and UTD was just down the street from us, and I didn't want to be very far away from my parents, so I went to university there. And UTD was kind of noted as a really great place for physics and astrophysics, which is what I wanted to do. UTD was founded by Texas Instruments, which is well known for making these calculators, you know. But it was also a place where a lot of the original research for the Apollo missions was done. So a lot of the engineering and space sciences work was done at UTD, mostly with contracts from the federal government. And at that time, it was known as the Southwest Research Institute. And it eventually became part of the UT system. So when I went to undergrad, I went there specifically to do physics, thinking I would go on to do a PhD in astrophysics. And I went through most of the undergraduate curriculum and figured out kind of near the end that I wasn't actually very good at physics, which is kind of problematic for a career in physics. And I went to an advisor who was a really great I think help to me, and he said, you know, have you ever thought of neuroscience? And I was like, no, frankly, I have never thought of it at all. And he's like, well, you know, we just started a new program here in neuroscience, and you're really good at kind of the electrical part of physics, not so good at the quantum mechanics part, which is what I needed to really excel at to do astrophysics. And he's like, you know, I think maybe neuroscience would be a good area for you. So I took a lab class actually as the first course, and I just immediately fell in love with it. It was like, this is what I was supposed to be doing my entire life. And I switched my major very quickly to neuroscience, and it delayed my graduation because I was already a long way along, but you know, that wasn't that big of a deal. And I started working in a lab almost right away, and it really was just a completely life-changing experience. And that is what got me interested in neuroscience generally, and specifically in neuropharmacology, which I'd say is the area where I kind of specialize. So that was how I ended up in neuroscience.
Let me tell you, you're so lucky, because back in India, because I did my undergrad in India, and it is so stringent that once you are selecting something, you're stuck. You can come out of it so easily, which is I love about US, that you can still change and you can still going physics to bio, like in neuroscience.
Right.
So it's not easy back in India, but it's great to hear the story. I hope a lot of kids can be motivated from this experience, because sometimes at this time when we are undergrad, we are not very sure about what we are going to do, and you realize once
Yeah.
you're back into that track that that's not right for you. So
It's so true.
that's a great story. So I know you were studying pain, and
Hmm.
when did you decide to study pain?
So that was an accident. um, I went to UT Health Science Center San Antonio, which is a terrific institution, like all of our health science centers in Texas are. And I went there specifically because I wanted to work with the chair of the department there, Alan Fraser, who's a terrific guy who does really great research. But his area of research is the role of serotonin in depression.
Mm-hmm.
And he played a big role, I think, in the early studies, kind of pinpointing the serotonin transporter as a potential target for antidepressants.
Mm-hmm.
He did some of the early work with Prozac, and really influential person in the neuropharmacology area. And I really wanted to work in his lab, and I went there pretty specifically to do that. And I did a rotation in his lab. So in the United States, in many PhD programs, you have to do rotations. And so I did a rotation in his lab. And while I liked everybody in the lab, it was a terrific lab, they were doing really cool research, I did not like the techniques that they were using. They were really difficult, long experiments. And I just didn't have the attention span or the patience for it. Yeah. And analyzing the data, like it was a little bit too qualitative for me, you know, coming from a physics background, and I just really wasn't comfortable with it. And I did a rotation with a guy named Ken Hargreaves and Chris Flores. So, but both of them together, I have to admit, like, I didn't really realize that I was working with one of the giants of the pain field at the time.
Yes.
But, you know, Ken and Chris were both such nice guys. So Chris is less well known because he went into industry, but he's had a fantastic career in industry developing drugs for a variety of different neurological indications. And he spent most of his career at Johnson & Johnson, but at the time he was in academia. And, you know, I just loved their lab. I loved the work we were doing, but, you know, I went there to work on cannabinoid pharmacology. And while they were doing it in the context of pain, I really didn't make a decision about it because of pain. It was because I was interested in cannabinoid pharmacology. And at that time, we were just making some of the early discoveries about how cannabinoid pharmacology worked, especially with the endogenous cannabinoids like anandamide, 2-arachidonyl glycerol. And it was a super exciting time to be in that field, kind of on the cutting edge. And I got the opportunity to do a lot of really interesting work, but it wasn't really until the end of my PhD that I decided to commit to being in the pain field. I'd actually applied for a couple different postdocs. One of them was to do pain work in Fernando Severo's lab at McGill. Another one was just a very general neuroscience lab, Oswald Stewart at University of California, Irvine. And then also Daniela Piamelli, who was a cannabinoid pharmacologist at also at UC Irvine. So, you know, I was really torn on what to do. And it was really kind of a coin flip, I'd say, on deciding which one of these to do. But, you know, ultimately I wrote a grant, got the grant, and when I got to McGill, that's where I think I really decided that I wanted to be in the pain field.
Wow, that's a great story. But let me tell you, like, about your mentor, Ken Hargreaves, because when I started studying pain, that's the first apparatus I used is to Hargreaves apparatus to see the heat pain, like general audience. So this is the thermal pain we're talking about, and Dr. Hargreaves made an apparatus which is like, you know, the most used apparatus for pain study. So yeah, you were lucky to work in the lab.
So, I was, no question about it.
So now coming to, like, we all talk about pain, and everybody's life has pain some way or the other.
Right.
But how do you imagine life without pain?
Well, life without pain doesn't really work. So, we
No, I
we
know, but
we
other scientists, yeah.
yeah. So I mean, we know that actually from genetic studies, right? So I think this is one of the most fascinating parts of the pain field, is that there are these congenital insensitivity to
Right.
pain genetic diseases or genetic disorders, and we've learned an incredible amount from them. So we have the first one that was discovered, which are the loss of NGF, nerve growth factor,
Right.
or mutations in TrkA. These people don't have these pain-sensing neurons called nociceptors at all. And from that, therapeutics that target the NGF system have been developed, and they might finally get approved by the FDA this year. They might not because of some side effects, but they're extraordinarily effective for different types of osteoarthritis, etc. And then there's the amazing story of NAV 1.7, which is this voltage-gated sodium channel. Yeah, it's terrific, that we have these loss of function mutations where people can't sense pain at all. We have gain of function mutations where people have pain without injury. And, we've learned a tremendous amount from that. And now we have these other mutations that are being discovered. So we have PRDM12, which is a transcription factor, which leads to loss of nociceptors. That one's extremely interesting. And now that there are all these other mutations that are being discovered that are maybe a little bit more specialized, but we had these voltage-gated potassium channels, for instance, very recently, that aren't really involved in whether you have pain or not, but allow people to have painless birth, which is amazing to me and amazing to many people, as I understand. And also this woman that lived most of her life without much pain sensation, who has a mutation in one of the cannabinoid metabolizing enzymes, fatty acid amide hydrolase.
Yes.
So it's a fascinating area of research.
Yeah, I mean, I remember when I started working on neuropathy, and at that time, because we are talking about NGF, nerve growth factor, and since diabetic neuropathy had insensation, like insensate neuropathy, right? So that time there was trial about injecting NGF to regenerate the nerve, so to improve the nerve, and the clinical trial failed when it came to I think phase 2. So I have seen the journey from injecting NGF for nerve growth factor and not having NGF, because NGF is causing so much of pain and inflammation. So
I suppose that trial failed because it actually caused pain in the people that they were trying to.
Yeah, and also the dose. I think we give humongous dose to animals, but when it comes to human patients, it doesn't work like so. It didn't work. And as far as the fatty acid amylase we're talking about, so I'm working on some of the gut movement, and this is one of the area I'm interested. So yeah, there is a lot of overlap in the field. With that saying, I will ask you that there is a saying called "no pain, no gain." We all know that, but how does it fit in your scientific journey? Like, what are your challenges you face?
There have been many challenges along the way, but I guess my single biggest challenge as a professional, a scientist, has actually been neuropathies that I have, and I'm very lucky to have had a good outcome. But I think my story with that is informative because it number one played a big role in I guess the empathy that I have for people that have chronic pain disorders, and number two, it became a big drive for me to try to develop better therapeutics, because I think we've done a really bad job of developing therapeutics, especially for neuropathies. Yep. So I had a back injury when I was a teenager playing football, American football, and I never really recovered from it. And when I was a postdoc at McGill, my lumbar spine basically exploded. So I had a really severe disc herniation, and the herniation went, I mean, it went on the outside, but a good portion of it went centrally. And I was paralyzed in my left leg. So, yeah, and you've seen me, like I'm not paralyzed right anymore, but it was quite
I
an
know you
event. I
had
mean,
the
I had,
injury, but I never
yeah,
knew that you were.
yeah. So I limp a little bit still, but it's nothing like what it was. And so yeah, I mean, I had a lot of pain on and off from basically the time I was 17. I still do now, but you know, it got really bad when I was in my late 20s, early 30s. And this disc herniation, for a couple months, I had just unbelievable pain. I really couldn't work. And one morning, I'd had a particularly bad night that night. It had been extraordinarily painful and couldn't sleep. I was taking a bunch of opioids to try to deal with it, and I woke up and I couldn't feel my leg. And yeah, so I picked up my phone. Actually, that's not true. The first thing I did was I tried to get out of bed, and I figured, you know, my leg's just asleep. And I fell on the floor because my leg didn't work at all, right? And I picked up my phone to call my wife, and then my wife's phone rang on the other side of the bed. She had gone to work and not taken her phone. So I was like stuck there on the floor in the house. So I called a friend, and he took me to the hospital. I don't know why I didn't just call 911, because I probably needed it. And went to the hospital, and you know, it took a little while to have the imaging done, but I needed a surgery to repair my spine. And I'm just lucky that I regained motor function. It was about 50-50 chance going into the surgery.
Where
So
were you that time?
I was at McGill, so I had surgery at McGill University Hospital.
Okay.
I was treated wonderfully by all the surgeons and the Canadian healthcare system. I won't go into how I probably would have been financially ruined if this would have happened in the United States rather than in Canada. So, you know, I was somewhat lucky that I decided to go to Canada to do a postdoc, given that this happened. But the thing that I will always remember from the whole thing is that, you know, that 2 or 3 months where I had really severe neuropathic pain going all the way down my leg, I was just completely incapacitated. I couldn't work, couldn't think, couldn't do anything. And I just remember thinking that if this doesn't stop, my career is over. There's no way I can work like this. There's no way I can do this job like this. And I think that I'm amazingly lucky that I recovered. I'm amazingly lucky I was in the position to get top-notch medical care from mostly people that knew me, because I was part of this big scientific group, you know. And a huge number of people don't have that kind of privilege in their life, and probably wouldn't have recovered from it. And you know, that really was a informative thing for the rest of my professional life, because it helped me to really understand better what chronic pain patients go through. I think a lot of people meet a chronic pain patients who say, you know, my leg feels like it's on fire all the time. But you're sitting there, how can your leg feel like it's on fire? You know, you do adapt, right? You can't be in this constant state of high blood pressure and your heart racing from being in pain. That's how it starts, but that really goes away over time. And it doesn't mean the pain is not still there. It's just, you know, your body has to adapt, or your body would probably not last very long, right? Yes. And the other thing is that I really realized at that point that the drugs that we have flat out don't work. So, you know, opioids, like I would take them because it was so unbearable, but they just take the edge off.
Yes.
And all the other things they do to your body, like you're studying gut motility, right? I mean, yes. you talk about stopping everything, you know, it's awful. And then, you know, I took gabapentinoids, and, you know, I was having a lot of trouble concentrating on anything, you know. But I mean, you combined the pain with the gabapentin, and I didn't even know who I was. No. It was insane. Yeah. And those were basically the option. If we tried an SSRI, it did nothing. Mm-hmm. Yeah, I can't imagine.
Yeah.
Dealing with that all the time.
Life is very different what we read in textbook for pain, right. For sure.
Right. Yeah.
So I know you said that so here comes the brain now. because pain is a perception that our brain processes. So I know you work on brain mechanisms also. So tell us about how you connect pain and brain.
Well, it was really that experience that I had with having such severe pain that I couldn't concentrate, couldn't really make decisions, that got me interested in this particular area of research. And one thing that's interesting about the pain system as opposed to our other somatosensory systems is that the other systems like touch pathway, the vision pathway, the sound pathway, they wire up kind of specifically. But the pain system, it goes into your brainstem. Part of it goes to your thalamus too, but it distributes to lots of different places in the brain, just on the most basic kind of inputs to the brain. And I think that tells us something about how pain really is like an alert system that is designed to activate lots of different circuits. That creates this emotional and somatosensory experience that pain obviously is, but when that goes on for a long period of time, it has a huge impact on lots of different areas of your brain. So we got interested over time in trying to kind of understand whether the brain would rewire or whether it would change neuronal phenotypes in different areas of your brain. We initially focused a lot on the amygdala, and I think the amygdala obviously plays a really important role in all of this, but frankly, there were a lot of other people working on that already, and we decided that we wanted to switch kind of over to the prefrontal cortex. And I think we've made some pretty interesting observations about molecular changes in neurons that we know are involved in cognition that are in the prefrontal cortex. We're limited to doing this in mice right now, but we work a lot with people that do functional brain imaging in humans. And I think there are some similar changes that happen in mice and in humans in the prefrontal cortex that are involved in executive decision making and just normal cognition that are they're not permanent. They seem to be reversible, but they're disturbing. Because it really demonstrates what a huge impact pain can have on many areas of your brain. And it's not just like there's an input there that makes the normal circuitry work in a slightly different way. It literally changes the circuitry, and I think that tells you just what a big impact persistent pain can have on your entire brain.
Yeah, I mean, I did some experiments on diabetic pain, and we initially, I'm a peripheral nervous system person, but I try to see what happens in the central, but not like you or Dr. Nigobar, who are looking at the amygdala or prefrontal cortex. But when I did that experiment, I have seen that the diabetic animals, as far as the cognition is concerned, they are not interested. Their brain is so full of the other things. Like if you see the normal animals, they're just going around and it's a new place, so they're exploring and all this. The diabetic ones, they don't want to move. They just moved a little bit, saw, and I'm done. I don't want to, like, that kind of thing. So I can see, like, people, if you see that in the people, like, people are so depressed, they don't want to come out, and so that affects brain. And you're right that the brain wiring gets different when you have pain.
Yeah.
So yeah, it's a big challenge. And so from there, so tell us about your journey from bench to bedside. I know you established a company, and I know you're helping people to somehow alleviate the pain.
Yeah.
So, tell us about that.
Yeah, so we, I guess I had this idea when I first started my lab that, we would make discoveries and then we'd work with people in pharmaceutical companies, and they would take these and then develop them. But I learned fairly quickly that wasn't actually the way that things worked, and that if we were going to be successful in taking our ideas and turning them into therapeutics, that we probably needed to do it ourselves. So I've now started 5 companies, and I really love this part of my job. Actually, I think it's the most exciting thing. I mean, individual discoveries, that we make in the lab still trumps everything. I mean, there's nothing more exciting than being the first person to know something. But one of the things that I really enjoy is thinking about how to take discoveries that we make and turning them into opportunities to commercialize it. And by commercializing, I don't mean necessarily making money. I mean turning it into something that can actually go through clinical trials and then help people at the end of the day. It probably will make money if it's successful, but the point is really to try to get new therapies into humans. And the first company that we started, called Searsci Therapeutics, that company actually just sold about 2 weeks ago. It is now part of Acadia Therapeutics. Yeah, so that's really exciting because we had the scientific and technical expertise to develop the molecule, do all the basic work that was needed. We also had the expertise, I think, to complete the phase 1 clinical trials. That did involve bringing in a lot of people that weren't involved in the company when we first started, and all these people worked together amazingly well. And there's a huge team that deserves a lot of credit for what we were able to accomplish. And I was just a small part of it at the end of the day. But still, we really have to work with another group to get through phase 2 and phase 3 clinical trials, because that's just such a huge operation. So having the opportunity to work with Acadia, I think is really going to help us tremendously, because they have the team in place. They're a mature company. They were founded in 1992. So about the time I was finishing my undergraduate degree, and they have a huge group that does just this. They do late stage clinical development. They have the capital that's needed to do phase 3 clinical trials, and we're going to start with post-surgical pain. I think that they're going to move into diabetic neuropathy. We have some really nice data that we've generated over the years for our lead compound in diabetic neuropathy, and then there are some hints that this mechanism might be effective for ALS.
Oh.
So I think that they're also going to give ALS a shot.
What are you targeting?
So the drug that we developed, so when we started Searsci, we were working on 2 things. So we were working on AMP kinase activators, which came from my lab. And then we were working on something else called reactive oxygen, reactive nitrogen decomposition catalysts. And those were developed by a medicinal chemist named Scott Dax, who was also one of the co-founders of the company. And at first, we actually got our first grant on AMPK activators, and that was all looking really great. And then all of a sudden, we got this big direct to phase 2 small business grant from the NIH here in the United States to fund our IND enabling studies. So that's the investigational new drug enabling studies. And basically that takes you from your preclinical work to phase 1. And that was such a huge deal for us that it shifted our focus from AMP kinase to the reactive oxygen, reactive nitrogen. And then that program is ultimately what we ended up developing. That's what Acadia bought. We never did develop our AMPK activator program much past the very beginning stages. And so what's really unique about the molecule that we developed is that it doesn't have the metal catalytic core that almost all of these other molecules have, and that dramatically improves its toxicology. So there are other reactive oxygen, reactive nitrogen, I wouldn't say catalysts, decomposition catalysts, but scavengers that are out there, but they all have this metal catalytic core. And giving somebody a molecule that has iron in it, you give them too much iron, it's not good for them. Sometimes it has manganese. It depends on the molecule. Ours doesn't have that at all. So Scott did an amazing job developing this new chemistry, and it just dramatically improves our ability to really have an impact on reactive oxygen and reactive nitrogen species. And we know with acute injury that that's one of the key drivers of acute pain, which is why we're developing it, at least at the start, for post-surgical pain.
I think that will work for diabetic pain too, because Diabetes.
I think so too, but I'm glad to hear you say that.
No, just hearing that, I'm just thinking that, like, I know that cup. That's from
You
Jack.
you gave me this cup. That's why I'm using it today. I was seeing how long it would take you to notice.
Yeah. So yeah. So I think, yeah, it's a very promising field you are working on, and good
Yeah.
luck to you for that.
Thanks.
So now, so from a scientist to an entrepreneur, that's a big way. And not a lot of scientists go to that path, or they're afraid to go to that path. And especially for the new generation, where a lot of people think science is good, but we don't make money. So, and this is a great example that you love your science, you do your science, but you still can go to the other path and satisfy your other needs. And a lot of people, I mean, they go to company just for that, but that doesn't satisfy their quest for whatever their scientific journey is. So I think your story is excellent, and I would love to hear some of that part of the story, and how you can really motivate those future scientists.
Right. So I think that this is a really great opportunity for people like us and young people in training who want to be scientists. I think entrepreneurial activities, especially in therapeutic development, are growing. And it's not easy to do, but nothing is easy to do. But it's not nearly as scary as it sounds. So first of all, starting a company is quite easy. I mean, I don't know the specifics of doing it in India, but doing it in the United States, it's very straightforward. There's some legal paperwork that you have to do. There are a number of rules that you need to follow, but that's easy and relatively inexpensive. I think what's hard is to know about kind of how to think about running a business versus running a lab. And I would say for anybody, they need to kind of think about how they want to do this as a career. So from my perspective, I don't want to give up running an academic lab. I love making discoveries, although I don't make them anymore. My students make them, but I love training students. I enjoy teaching greatly, so I don't want to leave the university. So for me, what that tells me that I need to do is that I need to find people that I trust to work with to start a company. And the way that I've done this is by working with students at the university that want to go into industry, but maybe they don't want to go work for a Merck or a Pfizer or something like that. They're a little bit more risk tolerant and they want to start their own company. And I work with them to put together the science part of the business plan. We have a really great program at University of Texas at Dallas, an entrepreneurial program that trains them on a lot of the stuff that they're going to need to do. And then, basically they start competing in like these business competitions, and that's a good way to get some early investment. We write grants, so every company that I've founded has started by getting SBIR and STTR grants, which are these small business grants from the National Institute of Health in the United States. I understand grants like this exist in most other countries, and it's a great way to get some non-dilutive funding to start your idea rolling. And from there, you just really need to focus. So every one of the companies that we've started has had pretty much a singular goal. They're developing a certain kind of compound or doing a specific type of thing. They're all in the pain space. And I think you just need to focus. There's, I don't want to say it's formulaic, but there's very specific things you need to do to develop a molecule from an idea to something that can go into a phase 1 clinical trial. And I think having a good deal of focus on getting those things done will get you to where you need to go. And then at the end of the day, I think the risk that you take on in starting a company is that you may find that what you've done is not going to work. And I think that you need to know at what point you would say, all right, this isn't worth pursuing anymore, and then move on to the next thing. But I think what I've learned in seeing people that do this, is that it doesn't really matter if you succeed or not. That there are going to be scientific reasons for you to succeed in one case versus not in another, and it doesn't have anything to do with how good your idea is. Just you don't know when you start discovering a new mechanism, make new molecules, you don't know if they're going to be safe or not.
Right.
That's why you have to do all this work. There's a lot of things you can try to mitigate along the way. You can de-risk as much as you want, but there's still a lot of risk involved. What people want to see is that you did a good job, you did your diligence all along the way, and if something gets derailed in the end, if it gets derailed because that there was a specific toxicology that nobody could have anticipated, nobody's going to fault you for that. It just is what it is, right? So if you do a good job, it doesn't really matter if you succeed or fail. Your record will show that you did everything the right way, and then somebody will gobble you up to do it again. So and people that work in this, they typically make more money, and the ultimate payoff that you can have if you're successful is gigantic. So, and I think once you've had that, then it opens up all kinds of doors to you, because you can start to fund your own things that you want, and you don't have to that you want to do, and you don't have to necessarily bring in all these investors who ultimately will reap more of the rewards.
I have a quick connecting question, like from your SBI grants and to go to the phase 1 clinical trial.
Right.
So what are the hurdles you got? Like, getting patients, make people convinced especially your, the medical counterpart, like, your,
right.
How did you convince them to get into the phase 1? Because that's the hurdle.
Yeah, really, from the perspective of the FDA, which is the big regulatory agency in the United States that makes the decision about this, there's just a lot of boxes that you have to check. We had to do long-term dosing studies in rats with a range of drugs. We had to know something about efficacy. And we'd done most of those studies, and then we needed to show like a difference between the efficacious doses and toxic doses. In our case, the molecules were extremely safe in rats, so that was quite easy to do. We had to do a large animal study, and almost everybody does it in beagle dogs in the United States. Our compound was safe there, and then we had to do a bunch of stuff related to manufacturing and how we were going to make the drug for the phase 1 clinical trial. That's totally outside of my field, and we brought in experts to do all that stuff. We had to raise money to pay them, but that was a risk that the investors took on, and it was it was all actually, again, pretty formulaic. The thing that was hard, I think, was the really early discovery part of it. Once you get to doing the IND enabling studies, it's basically a whole bunch of experiments that you need to do that are either going to succeed or they're going to fail. And you might find out that a molecule that you take into dogs is just flat out not safe. And if that's the case, you better have a backup plan in place, because it could be that the mechanism that you're targeting is not safe. If that's the case, you may not be able to take it forward at all. It could be that the molecule just has something with it, it creates a metabolite or something. Yeah. So you need to have some backups in place. And we just talked to as many people as we could talk to who had done this before about how did you de-risk your projects, and we tried to have as many of those things in place. And it ended up working out very well for us.
Awesome. So now we'd like to know who, not Dr. Ted Price, but who Ted Price is.
Well, I'm a dad. I guess that I would say dad and husband are the most important things to me right now. My kids are young. The pandemic has changed a lot of stuff for our family. So in some ways, it's been a gift because my kids are 2 and 5, so I'm at home, as you can see. I've been at home almost every day since March. In fact, I've been at home every day since March, and the reason is that my wife's parents live with us, and I don't want to call them elderly, but they are. They're in their late 70s, and they're a super high risk group, and they mean a lot to us. So we have done everything that we possibly can to avoid getting sick, and so we all stay at home. And it's been an amazing. It's frustrating to stay at home, but it's been an amazing gift to be with my kids all the time over this time. And I feel. I mean, you always feel a huge connection to your children, obviously, but it's been amazing to be with them every day, and I've loved that. So I would say, that's the most important thing about what we do. But I think every parent feels that way, is raising our children, right? But and yeah, I mean, I love being a scientist. I think this is absolutely the best job on earth. I love the flexibility that comes with being a university professor, and I also like to do other things. I used to play basketball a lot, which is I guess my big hobby. Can't play basketball right now because of the pandemic, but I'm looking forward to playing basketball again someday. I'll be even worse than than I am right now. And my wife is a nurse practitioner and also a scientist. So she's a nurse practitioner and PhD, and she has. She did trauma surgery in our huge hospital here up until the pandemic really got rolling, and now she works from home. She does occupational health, taking care of COVID-19 patients by phone. So these are people that aren't sick enough to be in the hospital.
Right.
And that's been amazing. I mean, we have 2 offices here, and so we're both working almost every day. And my wife is on the phone most of the day, so I hear, all these conversations, and it's been a real education to hear, her talking to patients who have COVID-19, and what's happened here in the United States. I don't know what it's like in India, but in the United States, it's quite out of control in many places. And
same in India too. Yeah.
Is it? Yeah.
Mm.
I am just scared to death about what the long-term consequences of this are going to be, because it's quite clear that people that get moderate to severe disease don't seem to get better on the time frame that we've had to experience this so far. And
people are getting reinfected.
yeah,
I have family members who have been reinfected back in
really,
India. And also the other thing I think is the inflammation, which people are not talking much about, but I think inflammation has a big role here. And we all know that in pain also inflammation is one of the biggest thing we have to reduce.
Without question.
Yeah.
Yeah.
So I think COVID has that part also needs to be explored someday.
Yeah. Yeah.
But it's nice to hear that, and I could see from football, American football to basketball. So at least your surgery went well. Now you can leave.
I definitely can't leap. I'm very stuck to the ground, but I can shoot from a long way out, which is a good skill to have as you get older in basketball. Yeah, so that's my primary basketball skill. I'm definitely a 3-point shooter. Yeah. Yep.
Awesome. So I think I'm so happy to talk to you. I'm sure whoever is listening has got a lot of information, but I would open this to questions if anyone has questions. And maybe through the chat window, or maybe some of the panelists can come back up.
Thank you, Professor Theodore. It was quite enlightening as well as inspiring for the young research minds to understand that we can have research works which can have therapeutic applications. Understanding the molecular mechanisms that causes the pain to be chronic, as well as how the brain neuronal plasticity alters the way nostotic informations are collected from the sensory neurons, and how this is related to chronic pain in human patients was quite interesting to watch. With the discussion between Dr. Momun Chattopadhyay and Professor Ted Price. Thank you, both of you. May I now request our Honorable Vice Chancellor, Ravenshaw University, Professor Ishan Patra, to kindly address the session.
Hi, Ted. Hi, Munmun. It was a wonderful discussion, and it has given so many things. You started with your love for your own land, and then to your work, to the present day issue. I'll come to the lecture later on, but there is one question that has come up. Let me put that to you first.
Mm-hmm.
It's a very simple question, though. When we are testing any new molecule or drugs, especially for brain, the choice of experimental animal becomes very important.
Yeah.
How are these experimental animals chosen for pain research?
Well, that's a great question, and actually an area of a lot of controversy now. So, I don't know what the answer is. I don't know if there's any one right answer, but I can tell you how we're trying to address that. So, essentially, my entire career, I've been concerned that we were potentially using the wrong species to study what we were trying to study, and it's always been. So I remember when I switched from physics to neuroscience, which we talked about earlier, that I was kind of blown away that at the time almost everybody was using rats for their studies. I didn't know this going in, and then went into a research lab and I was like, why are there rats everywhere? Why are we doing everything in rats? And I guess I understand why we use the species that we use now, but it's still a good question about whether we're making the right choice from the perspective of therapeutic discovery. So I'll tell you how we approach this now. So we try now to do as much as we possibly can on human tissues. So I mostly study the peripheral nervous system. We talked about what we work on in the brain, but still, I mean, for drug targeting, we mostly are trying to target the peripheral nervous system. So we have developed collaborations with neurosurgeons and also organ transplant surgeons, where we go into these surgeries or organ harvesting, and we take the peripheral nervous system from the organ donor or from the patient and do molecular profiling experiments, mostly using RNA sequencing, but increasingly doing it with proteomics and also some signaling assays. So we try to start with seeing whether the target can be manipulated in the human nervous system. Then there are obviously things that we're not going to be able to do in humans that we want to try to assess behaviorally, etc. But we can do some of these same molecular profiling experiments in mice, rats, and also pigs, actually, we're using now. And we can look to see if for instance, we're trying to target a G protein coupled receptor. If that G protein coupled receptor is expressed at the same level and the same type of neurons across these different species, because we have some pretty good pre-existing information about what it looks like in humans. So we can then make a choice about which species to use. And if it turns out we can use all three, mice, rats, and pigs, I can tell you for sure we would choose mice because they're the least expensive, the smallest, etc. But there may be targets, and we've actually found one recently, where we're pretty sure that we cannot study it in mice and rats because it's not expressed in mice or rat sensory neurons, but it does seem to be found in pig sensory neurons. So those are going to be some expensive studies for us to do, but we do think it's a good target, and I think it's worthwhile. So we likely will pursue that, but we're not going to be able to do it in mice and rats. So I think now we have so much information about this kind of thing that we can make those kind of decisions. And our approach to it is trying to start with the human and then make a decision about what kind of model to use.
Yeah, I believe Supriya Das has got the answer for that. There's another interesting question. I'll also add something to that. This comes from Somerajan. He is asking why the pain at different organs are felt differently, although all are processed by brain. And
Hmm.
what I would add is why some of the pains become real great pleasure, while while pain is after all pain.
Right. Well, I mean, I think that the primary reason that pain from different organs is felt differently is because the nociceptors that innervate those organs are wired up to the central nervous system in different ways. So, for instance, areas of your skin um, wire up to the dorsal horn in a very specific way, whereas pain from some of your organs is more diffuse. And also, you have inputs coming from two different areas. So, you have inputs that come in through the dorsal root ganglia to the spinal cord, but you also have nociceptors in the vagus nerve that go up to the brainstem. So, one of the biggest things about having visceral pain versus somatic pain is that somatic pain is usually very easy to localize, whereas visceral pain can feel like it's coming from like an entire side of your body. And a lot of this has to do with just the basic neuroanatomy of how they're kind of wired up. Um, certain types of pain can, or certain types of activation of nociceptors can be pleasurable, as you just mentioned. That seems to happen also because of neuroanatomy. So we have these certain nociceptors, certain types of low threshold mechanoreceptors that actually mediate like pleasant touch. But if you have a nerve injury, that pleasant touch can go from being pleasant to being very painful, which we call mechanical allodynia. So, again, it kind of boils down to neuroanatomy. In terms of pain receptors, we have an amazing diversity of receptors that are expressed by these sensory neurons that innervate our body and our organs. And those, some of them we understand extraordinarily well. So we started today talking about TRPV1. TRPV1 we understand in minute detail. Other ones we have very little information about, and we don't know which specific types of sensory neurons express them. So I think this is an area for huge opportunity, trying to understand, what are the mediators that are released during inflammation and how do they act on different subsets of nociceptors. Is still an area of unaddressed need.
One of my colleagues here, Dr. Dhananjay Soren, has got a question: how temperature modulate the regulation of small molecules in pain sensation in spintomatic themic tract of brain?
Could you repeat the question? I didn't, that was a
There
complicated
is
question.
how temperature modulates the regulation of small molecule in pain sensation in spinothalamic tract of the brain.
Well, I mean, temperature acts on specific receptor or different ranges of temperature act on specific types of receptors. So, for instance, we know, again, going back to TRPV1, that TRPV1 is activated by noxious heat. TRPV2 also seems to be activated by noxious heat. There have been some other trip channels that have recently been described as responding to noxious heat. Noxious cold is activated by, or activates a different kind of receptor called TRPM8. So there are specific receptors for different types of temperature, and those different receptors are expressed by different subsets of neurons that innervate the central nervous system differently. So they activate presumably they activate different types of neurons that go into the spinothalamic tract, and that's why you get different percepts from activating those different neurons.
Yep. So that if some more questions comes, then I'll come to that. And it was a very wonderful discussion between Munmun and Ted. And if you know, Munmun was my first PhD student to submit her first PhD thesis with me.
That's
And
great.
she did her work when we were just developing the lab. So she
Yeah.
knows the pain of developing science and doing
Yeah.
research in developing group. And you will be happy, Munmun, with the new education policy of India. Now, in a few years to come, even Indian students can move from one area to another area. Not only in science, they can move to commerce, and commerce student also can come to science. That's what is targeted.
It's good to know.
Yeah, this is something that has been targeted like that, and in the early periods, of course, not during specialization.
Yeah.
So it's the right thing that that Munmun said, and you also agreed, and I put it like this, that well, there is no pain, no gain, and if there were not painful happenings with you, you'd not have thought seriously about working in pain, perhaps, or maybe your feeling for people that you are going through, that more people should not go through, that has brought you
Yeah.
to identify molecules, bring them to from your bench to the bedside.
It's
That's a
very true.
very great and very interesting thing. And I also appreciate the way you said pain in brain. Actually, you can't think leads to thinking the issue about the pain itself.
Yeah, that's true.
Pain stops you from thinking, so you start
It
thinking
did.
about
Yep.
it. So, and uh, the way you have trodden the path from discoveries to products to human use to earning money. At the end of the day, everybody needs money also. And if
Yep.
you have done something, you must be paid for that. The society must pay back your efforts that you have done. And it's a good message that you have given to the students, the young faculty and young scholars here, that love for science is one thing, but to work with a focus is more needed. And then
Mm.
the third thing and most essential part is to finally decide to earn big money too. That should be the way, you to complete the story. And actually it is very difficult for one man to do that, and Ted, you have done it, so it's a big applaud for you. And we must say that's a great job done. Not many people
Thank
can
you.
do, starting from suffering to finding out a really good treatment for that and therapeutics for that, and then getting the therapeutic translated into a product which can be uh, sold out and earn money on that. And this is something that's a great motivation for the young Indian scientists listening to you. And I'm also happy that there are many young Indian scientists who have started thinking like this, and they've gone into the startups and things like that.
Yeah.
And it's very nice that the way you say it, don't lose, and if you lose, then take the risk to raise again. That's the very,
Yeah.
very nice message that we have got from your lecture. And at the end of it, really, the way you said, I am also facing the same thing. There's so many things to do as the leader of an educational institute, but so little that you can do outside your home. So I am also put to home and put to my office room only. And so many things that I
Mm.
can do. I am not able to meet my students, and for a education leader, not meeting the people who are really the beneficiaries doesn't really help you and doesn't keep you happy.
It
But
is
I'm
hard.
I'm also happy that my daughter and my granddaughter and son-in-law all have moved to this place and they are with me. It's definitely true that COVID is, of course, given it to a price, but COVID has helped us to connect. We have now understood how to connect and think. If I
Yeah.
wanted to have stayed in my university, it would have not only needed money, but it would have needed a lot of time from TED to come and spend with and to give this lecture to our students and the faculty. And I would not have ever known that my very first scholar has become that mature of holding a conversation that good. There's something. It all gives. It all has come with COVID, and for Indians, what has happened. This is a blessings in disguise one way, that we have understood how to communicate easily with the use of modern technology, and
Hmm.
we are using it very often. And people in India are used to this system now, and even people who had uh, with very little entry into computations or computers or laptops have started using this for this mode of communication with their near and dear ones, and then telling their own part of story to people.
Mm-hmm.
So in by and large, it was a very interesting uh, lecture, I mean, discussion that we had. There are a couple of questions that have come up from our colleagues. There is one, it says, when we experience happiness, sometimes tears comes out of our eyes. So also during pain. What is the relationship between these two?
That's a good question. I don't actually know the I mean, it's obvious that happens, but I don't know the underlying neurobiology, and I don't know that anybody does actually. I mean, I think it just illustrates how intertwined the pain system is with our emotional centers. I mean, we know that the pain part of the somatosensory system distributes throughout all of the brain, but I mean, it just goes to show what a huge impact input from that system has on the emotional part of your brain.
See, the most interesting part of this Ravenshaw 150 lecture series is that we have been able to attract all walk of life, all kinds of specialists to listen to each other. I've got a colleague from the Odia department, which is the regional language here. They speak the language that we speak in Orissa, in India. And she has got a very interesting question because she is a lady of literature. Her question is: we get physical pain and sometimes emotional pain.
Mm-hmm.
Are identical brain regions responsible for both the kinds of pain, or different reasons are involved in it?
Yeah, that's an area where a lot of brain imagers have done some really interesting work. And I mean, I think what we know is that some of the brain areas are shared, but there are other ones that are distinct. So, some of the networks that are activated that are involved in emotional pain have the same kind of impact on your ability for cognition, etc. But then again, the somatosensory part of it is not really present, and some of the kind of protective circuits don't get engaged. So while I think emotional pain obviously has a very big impact on your brain, that pain that involves the activation of nociceptors in the peripheral nervous system ultimately has a bigger impact because it activates basically all of these circuits at the same time.
Good. So this has been a very interesting uh, lecture in this, I mean, discussion in this series, and I'm pretty sure that this has been a successful one because of the last question I got. Uh, this has attracted really people from all kind of walk of life, and I'm so happy, Ted, that you accepted my request and spend time to be with us and talk to us. And
Sure.
now there's a sort of a formal vote of thanks from our students and from our young scientists. I'll pass it on to Luna and her team.
Okay. Thank you. It was a pleasure. It was really a pleasure to be
Well,
with you guys.
I just want to make one mention here. Like you
Okay.
said, when we were in your undergrads, and you didn't know really why to study neuroscience. When we introduced the masters in neuroscience in Jivaji University, that's the first ever department of neuroscience in this country. People all started asking me the same question: why neuroscience, and why that specialized subject? And now uh, I'm very proud to have started that program because now everybody, every walk of, every specialization, students from every specialization wants to come and study neuroscience, from psychology, from
Yeah.
dentistry, from medical science, and from biological sciences, from all areas. And one of my colleagues, who is a professor of economics, he has become tremendously interested in neuroeconomics and things like that. People are really getting so. It was a wonderful starting you had to land up here, and I wish you all the best, much more success and feathers to your cap. And I can see a small basket at your back.
Yeah.
And you keep playing on that also, or it's just there?
With the kids. With the kids.
Yeah,
Yep. Yep.
okay, okay. So all the best, our respect for your parents and love for the family. So I pass it on to Luna.
Thank you.
Thank you, sir. Actually, us on behalf of our student, my student Dr. Lilipta wanted to put the formal vote of thanks to Professor Tate and Dr. Munmun. So it is over to Nilipta.
Distinguished guests, respected speakers of today's webinar, as well as all the participants, I like to acknowledge the put forth my formal vote of thanks to today's part. We have Professor Theodore Price as well as Dr. Mohan Chattopadhyay, who, with an innovative way of discussing all the topics, covered all the topics of chronic pain as well as the various mechanisms related to brain neuroplasticity. It was quite a break from the monotonous one speaker lecture series of this webinar. So thank you both of you for sparing your valuable time with us. I would also like to acknowledge the support and contribution of National Academy of Sciences India, NASI, Institute of Life Sciences, ILS, and Indian Academy of Neurosciences, IAN, in organization of this webinar series. Last but not the least, my heartiest gratitude to all the participants who choose to be live. As well as the Vice Chancellor, Honorable Vice Chancellor, Professor Ishan Patra, who for organization of this webinar. And so lastly, I would like to put forth heartiest thank you to all the participants who choose to be live with us today and contributed in making today's webinar a successful effort. Before we end, I would like to add that tomorrow we have a lecture by Professor Renu Wadhwa from National Institute of Advanced Industrial Sciences and Technology, Toshiba, Japan. She would be talking on ashwagandha for stress, health, and COVID-19 management. The talk would start at 4 p.m. tomorrow, Indian Standard Time. 4 p.m. Indian Standard Time. Thank you all.